Encorafenib, Binimetinib, and Cetuximab in BRAF mutated colorectal cancer. Definition: patients with metastatic colorectal cancer with BRAF V600E mutants have and poor prognosis, with a median overall survival of 4 to 6 months, and after failure in initial therapy.
Basically, the BACON trial was designed to find new treatment options in patients with metastatic colorectal cancer that bear the BRAF V600E mutation. And we know that this accounts between 8 to 10% of the patients with advanced disease, and also we know from previous series that these patients actually have a poorer prognosis compared to the global metastatic colorectal cancer population. So the initial data suggested that as these patients had BRAF mutation, actually they could be sensitive to BRAF inhibitors, like for example patient with melanoma. But actually with the initial data, we realized that the activity was really very low, almost inexistent. And then we tried to understand in the lab which were the mechanisms of immediate resistance to BRAF inhibitors, and we realized that there was an immediate upregulate of the epidermal growth factor receptor, EGFR. So then we went back to the clinic and we decided to do the BACON study, that it's a randomized study in patients with metastatic colorectal cancer that have been previously pretreated and that bear the mutation B600E, BRAF B600E. So we randomized a total of 665 patients in 3 arms. One arm containing a triplet, a RAF inhibitor, Encorafenib, a MEK inhibitor, Binimetinib, and an EGFR inhibitor, Cetuximab. The second arm, the doublet, only contained a BRAF inhibitor, Encorafenib, and EGFR inhibitor, Cetuximab. And finally, the control arm was what it was considered a standard of care, and this was either irinotecan with Cetuximab or FOLFIRI with Cetuximab. And as initially expected with the preclinical and early clinical data that we had, the triplet combination did much better than the control arm. So the hazard ratio for overall survival was 0.52.
Applications is nurses. Patients receive Encorafenib, Binimetinib, and Cetuximab and triplet therapy group, or Encorafenib and Cetuximab and dual therapy group, or take choice of researchers and for Cetuximab and erotic or Cetuximab in FOLFIRI.
So for BRAF mutant, there is a big change, okay, because multiple studies showed that triplet combination with a BRAF inhibitor in the center, with a MEK inhibitor now, before it was also tested with PI3 kinase inhibitors, and an EGF receptor inhibitors, in this case always Cetuximab, showed very promising results. In refractory patients, response rate over 30%, which is unheard for this disease. Now, recent data showed at World GI showed not only high response rate, but long progression-free and overall survival, which is the reason now the new concepts are that there is a trial being developed in first line. So now the interesting thing is this triplet has no cytotoxics, only target agents, which again indicate when you are smart in identifying the molecular genetics of the tumor, you
Pros and cons. BRAF inhibitors are only limited activity due to reactivity of the pathway through signaling or takes epidermal growth factor receptor.
management of patients with this particular subtype of colorectal cancer can be difficult because of how aggressive the cancer can be. We're fortunate that we now have regimens that are giving responses for patients. The activity that we've seen in patients getting treated with the Binimetinib, Encorafenib, Cetuximab regimen have been encouraging. And I think what we've seen are patients that that can respond, tumors respond, can have improvement in their symptoms. And I think importantly, increased durability of their disease control, more so than we have seen previously. And that certainly provides hope that this is a regimen that can ultimately result in kind of approval and availability for our patients. Obviously, we'll have to wait for the phase 3 study to read out, but we are encouraged that the FDA has recognized this regimen with a breakthrough designation last fall. So they'll be rapidly reviewing the data as that becomes available. So for managing patients with BRAF V600, in particular with the Encorafenib, Binimetinib, Cetuximab regimen, it does require monitoring for eye toxicities. There can be with the Binimetinib a kind of transient blurred vision after dosing, but that usually resolves fairly rapidly within an hour or less of the dosing. But working with an ophthalmologist is sometimes needed if those symptoms become more problematic. Overall, the regimen is well tolerated.
ISIGO applies to Ecuador in this open-label phase 3 trial, where recurrent 65 patients with BRAF V600L metastatic collateral cancer who had disease progression after 1 or 2 previous regimens.
Thank you, Axel, and I appreciate the opportunity, and I apologize that you'll see me up here a few times, but I'll also note that I'm here for the updates, not for the debates, which probably is a testimony to my lack of persuasion, perhaps. But despite that, I'm going to try to persuade you about the relevance here of BRAF V600E mutation. And what I want to do is really convince you of two things. One is that this is a very aggressive subtype of colorectal cancer. And the second is that we actually have therapies that have now moved into clinical practice, and we have opportunities to continue to enroll patients in clinical trials and improve outcomes. So this is the 50,000-foot view, but what I'll be focusing on is really the BRAF mutated subgroup here. So the first point is to reiterate that when we're talking about BRAF, what we're in this context, what we're talking about is the most common mutation, which is the V600E mutation. This is an activating mutation, same one that's in melanoma, some lung cancers, some thyroid cancers. This is a mutation that makes the enzyme constitutively active. Now, as an aside, and I'll touch on this a little later, time pending, that there are other BRAF mutations that are of mixed activity, and some of them are inactivating. And these actually, as Axel has shown, this actually is a group that has better prognosis. So when we talk about BRAF, one of the key things
Conclusion: the safety and tolerability of the Encorafenib, Binimetinib, and Cetuximab regimen is manageable and acceptable of onset, and to establish this regimen as new standard of care of previously treated BRAF B600X mutants and CRC.
So the main argument that I would make, and I'll walk you through some key studies, is targeted therapy with just triplet combination should be what should be considered for our patients with metastatic BRAF V600E mutant colorectal cancer, and I'll show you why. So in terms of I just have one slide, and I'll walk you through all key studies. So this is a story of precision medicine, so bear with me. So here I'm showing you data which has led to why FOLFOXIRI with BEV, which Dr. Engel argued in favor of, has been the standard of care for first-line treatment for patients with BRAF V600E mutant cancer. But on the columns on the right, what you're seeing is how precision medicine has truly evolved for this subset of patients with BRAF V600E colorectal cancer. Often, in the world of individualized medicine, the naysayers would say, well, the BRAF is not a real target because the BRAF in melanoma, the same BRAF doesn't behave the same way in colon cancer, and that was truly the case over the last decades. As Dr. Van Kusten this morning pointed out, the studies with BRAF inhibitors alone, the studies with BRAF and MEK in combination, while it almost doubled the responses and survival in patients with melanoma, and even earlier this year, data in cholangiocarcinoma showed the same results. However, for patients with colorectal cancer, it never showed anything. And the reason for that is that on the RAS-RAF pathway, the reason why the RAF inhibition or the MEK inhibition, even a single agent or together, didn't work is upstream there is upregulation of EGFR, and what happens is the pathway is bypassed through downstream upregulation of CRAF. So what was needed was upstream blockade of EGFR. So you needed to block the pathway at 3 different.
Thank you.